So I am going to talk about circadian biology, which is the chronobiology, or which is also called as the science of the day night, the light and no light in our life, or the science of sleep, which is so closely associated with our metabolic physiology. The NAFLD, or the non-alcoholic fatty liver disease, is what I'm going to talk about, and also the neurobehavioral and epigenetic perturbations that some of the recent observations have been done. The most of this study has been done as a part of my SERB project, which I will be acknowledging at the end. So to begin with, yes, very proudly I am an MSUI, and very proudly I flaunt my university on any stage I give a lecture in India or abroad. So you can see the old Baroda College building, which is the Faculty of Arts, and right now it is under renovation. So after the renovation is over, I will show the renovated picture, which will, I'm sure, look very beautiful. The Maharaja of Baroda, Sir Sayajirao Gaikwad III, was a great visionary who brought fantastic scholars in various fields and disciplines to the Baroda College at that time in 1887 onwards. And none other than Sri Aurobindo Ghosh, a great visionary scholar and now even worshipped and revered for his philosophy, was once upon a time the principal of Baroda College. This is my university department, the Faculty of Science building. The ground floor is chemistry and the first floor is zoology. And on your screen, the extreme right side corner is where my lab is situated. And before I start my talk, all the people who are there online, I extend my warm invitation to one and all. You are welcome to visit the MSU campus whenever you are in Baroda, and please do visit my lab. I'll be happy to host you. So talking about the circadian basis of disease, now there have the fact that there is a circadian basis for lifestyle disorder is very well known. The circadian basis for the diabetes, blood pressure, circadian basis for insomnia, the food intake getting affected, your locomotor function getting affected, is all very well known. Now even people are talking about the circadian basis of cancer. So circadian biology, so to say, is into multiple facets of our disease biology. So far humans are concerned, not only the humans. I am in the department of zoology, so a lot of non-human primates, the lower vertebrates as well as the invertebrates, all show a circadian cycle. Some of them are the marine organisms show what is called as a circatidal rhythm, which is associated or rather synchronized with the low tides and the high tides. Not going into details of that, I'll just focus on the circadian biology. This is a beautiful slide which shows that at different times of the day and night, we witness different, uh, you know, physiological conditions. Like the deepest sleep is at night 2 p.m. Then at 4:30, our body temperature is the lowest, and at approximately 8 o'clock at night, 7:30, 8 o'clock, our body temperature is the highest. Likewise, there is a sharpest blood pressure rise early in the mornings, giving also the idea why lots of deaths are due to heart attack are there in the sleep. Melatonin secretion stops once the daytime, the sun rises, and there is high level of alertness 10 a.m. onwards. So any students online, if you have attended the classes before 10 a.m., your concentration levels are less. So you can always blame that on your melatonin levels. The best coordination is at around 2:30, 3 p.m. The fastest reaction time is at around 5:30 p.m. is what the time when most of the people go for play, outdoor games, and they people like gymming, and approximately 17 hours is the best time to do cardiovascular exercises and muscle building. People who are fond of gymming and all. Then talking about the blood pressure, the highest blood pressure is at around 637, and melatonin secretion starts peaking is by around 21, that is at 9 p.m. 9 p.m. is the time when we should retire to bed ideally, but with our changing lifestyles we are not able to sleep even past midnight, and there the problem starts. Here I am not touching upon, but just referring to the blue light which is emitted by the devices like the cell phones or the laptops, which disrupt the melatonin rhythm in our brain, in the pineal gland, causing problems. So the term photic Zeitgeber and non-photic Zeitgeber essentially refers to the suprachiasmatic nucleus and its control and its regulatory functions, wherein there is the central clock in the brain, in the suprachiasmatic nucleus, in the case of mammals, which detects light or no light through the eyes and, of course, other sources. There are also multiple peripheral clocks operating in the bone, in lungs, in heart and liver, that is my target tissue. And on the right side, that is the B, as you can see, is a the transcriptional translational feedback loop that the clock genes are functioning. Now here, you have to understand, molecular clock comprises of two key genes, which is the BMAL1 and the clock gene. So BMAL clock heterodimer is the one which will be switching on a lot of other downstream gene pathways, and that in turn regulate a another level of genes such as the per, that is period, cry, which is also called as a cryptochrome. So you've got per1, per2, cry1, cry2 and cry3, the isoforms, and there is the reverb alpha and the roar alpha. So these all are the ones which are in synchrony controlling the circadian cycle. You can read more about the transcriptional translational feedback loop, which is so much an core or the essence of chronobiology. Now, coming back to the melatonin, which is secreted from the pineal gland and it is bathing the entire body. Melatonin is a powerful antioxidant. Melatonin is having free radical scavenging properties, and melatonin, because of all these credentials, is also known as the youth hormone. So I, as a person who specializes in endocrinology, is always fascinated with the action of melatonin and in what way melatonin can be regulatory, or if I dare to say, have corrective functions in the metabolism of our body. Here, I wish to introduce you to, well, obviously on the extreme right, there are multiple metabolic functions which are under the purview of melatonin. There are the hormones. Glucocorticoids and melatonin are always antagonists to each other. Corticosterone levels rise is why we wake up, is the time when melatonin level have to come down. As the melatonin levels rise, the corticosterone levels have to be down. Only then we are able to have a peaceful sleep. Disturbed sleep is courtesy stress, that is high levels of corticosterone. Persistently high levels of mild stress are the basis, because melatonin secretion becomes weak, its action becomes feeble. The bathing of tissues as it is able to do at the night time, it's not able to do. Likewise, the eating, the behavior, the sleep-wake cycle, and in the long term, the obesity is also courtesy the levels of melatonin. This is a cartoon which represents the dark phase melatonin release and its action via suprachiasmatic nucleus. So, all in all, here you can see there are the daytime and the nighttime. What I have missed out possibly in the previous slide is melatonin is a tryptophan derivative, and melatonin gets released into the bloodstream, and it will be from the pineal gland, obviously, and it will be bathing the entire body, all the tissues. Uh, the melatonin also, besides having cytosolic receptors, have also got the surface receptors, the MT1, MT2, and a heterodimer of the two, and it is manifesting the desired effects. Now, talking about the disease which I am referring to, the non-alcoholic fatty liver disease, or also referred as NAFLD, which easily progresses to NASH, also called as a NASH, right? This is known, has been now called as a silent killer. Silent killer because, uh, as we all know, that the basic physiology that we have studied is the SGOT and the SGPT levels, or the AST and the ALT levels, which are indicators of the liver function, also known as the liver function test. The AST ALT levels, SGPT especially, it becomes very high, indicating liver damage. The problem is when the SGPT levels are high, detectable in the human body, the damage that has taken place as a result of fatty manifestation is reached a level which is beyond repair. Here, let me also remind you that liver is having a fantastic ability for regeneration, but this damage is so severe that it has progressed to what is called as NASH, or non-alcoholic steatohepatitis, and this the progression often takes place through overlapping events, through first hit and second hit. In first hit, there is insulin resistance that develops, which can be a condition called as mild to slightly severe diabetes. Is the second hit when the gut becomes leaky, the oxidative stress is tremendous, there are pro-inflammatory cytokines which are in the body, and there is mitochondrial dysfunction, which causes death of hepatocytes, and overall efficiency of the liver goes down, leading to NASH, or non-alcoholic steatohepatitis. What will happen if the liver is left unattended? It will progress to hepatocellular carcinoma. This is the link or the connection between the two. Uh, some update is that now NAFLD is called as metabolic dysfunction associated liver disease, or MASLD, and the another new term that has been used is, it is called as MASH, that is metabolic dysfunction associated steatohepatitis. The changes have been done for certain specific reasons to explain and incorporate the wider aspects or multiple aspects of newly found or newly discovered associated etiology of this disease. So talking about the experiments, I will start with a very simple experiment. Here you take cells in culture, you take the HepG2 cells, which are hepatoma cells, you feed them with oleic acid. Now the moment you feed it with oleic acid, they will accumulate oleic acid and they'll become plump. Now this is a representative in vitro model for a fatty liver. Now here what I do, I add melatonin and I see that the accumulation of oleic acid is much less as compared to without melatonin. That is what you can see over here. The accumulation is much less. Likewise, if I do a DCFDA staining, which is showing a bright fluorescence as a result of intracellular oxidative stress, the indice is much low if in the presence of melatonin. Now, this simple continuing further, the measuring the mitochondrial membrane potential with the JC1 stain also shows that there are less a higher number of J aggregates as compared to the, which is comparable to that of control. You can see here, if you can see my cursor moving. So this is comparable to control in the presence of melatonin. And as you can see here, it's evidenced by the fluorescence also. Uh, these findings, are trying to imply towards one thing, that melatonin is somehow able to prevent fatty manifestations in liver. I did not do anything new over here, but I have just tested the hypothesis, which is already known. But my curiosity was, my study was, can I subject the mouse to a photoperiodic manipulation induced chrono disruption? I refer to the circadian biology or the chronobiology, which is the day-night cycle. So it's the chrono disruption. So if I disrupt the circadian cycle by shifting the photoperiod periodically from Monday, Tuesday, Wednesday to a early day, starting much early, and which accounts to a phase advance, and restoring it back to the original one, which accounts to the phase delay, as you can see here, this will cause what is called as a photoperiodic manipulation induced chrono disruption. This is also referred as a jet lag protocol. It was published in 2016 by Kettner, and after that my lab has published multiple papers adopting this protocol with minor modifications in C57BL6J mice. So what have we done? We have subjected the photoperiodic manipulation induced chrono disruption protocol, and on the other hand, we have also subjected another group on high fat, high fructose diet. Okay, high fat, I repeat, high fat, high fructose. High fat diet, burgers and the pizzas, which are loaded with cheese, and high fructose diet, which is the cola, which contains high contents of fructose. So then we have manipulated the photoperiod and to see what happens. Here, from eighth week onwards, we have given melatonin intraperitoneally to see whether melatonin is able to alleviate the symptoms of fatty liver. Now, the question is, most of the studies start the treatment simultaneously. We started it somewhere halfway through the study, as you can see here. This is deliberately to mimic a therapy like condition where a disease actually develops and the person starts taking medicine and for a long period of time, and then we assess what is the effect on body circumference, liver weight, and all the metabolic parameters and profiles. There was an improvement in body weight gain, as you can see here, as a result of that melatonin treatment. The liver function markers, which were distorted, I'm referring to the AST and ALT, which were distorted both in the high fat, high fructose diet as well as in the jet lag, they were improved as a result of the melatonin treatment. As you can see here, the serum lipid profile also showed an improvement. Okay, the and these are the transcripts of genes regulating the lipid metabolism. They are that is the CPT1, PPR gamma, SREBP1C, which normally any person investigates while studying the progression of a disease which is related to lipotoxicity. These all were undergoing a change as a result of high fat, high fructose diet, the jet lag, or a combination of the two. Now, this is showing the visceral adiposity and the changes in the liver. So, this is the mouse, and you can see here very, very high levels of visceral adiposity can be seen over here. And the melatonin treated liver is showing a color which is similar to, almost similar to that of control. Well, exactly, it could not revert. Then the microscopic evaluation of liver also showed an improved melatonin profile. As a result of melatonin treatment, the liver was much better. There are certain parameters like the hepatocyte ballooning, which also is showing an improvement as a result of the melatonin treatment. Now, as a circadian biologist, we study what are called as the oscillations of the genes. Now, oscillations of the genes are nothing but a time point based study where the samples are collected in the daytime as well as in the night time. Now, this dark box which you see on the right side of each of the graphs is the one which is representing the nocturnal period, whereas the brighter one or the white one is indicating the diurnal period. So the clock gene, the BMAL one, the PER2, and the reverb alpha, each of them you can see that the clock peaks in the daytime and the clock has got a nocturnal peak also. The BMAL has got a diurnal peak as well as a nocturnal peak. Per peaks sometimes in the evening and stays higher till midnight, whereas reverb alpha is peaking just before it gets dark. So this is the characteristic when it comes to the peaking of the genes in the liver tissue. This is what we had observed. Here, the peak of BMAL in an early, in the day phase is indicating that our control animal is having perfect circadian rhythm. Likewise, the clock showing two peaks also was there. The PER is also showing, the PER1 and the PER2 also showed, but the CRY2 showed a ebb as compared to the other genes. By giving high fat diet, you can see here that there is a decrement in the peak. All right, there is a decrement here in the peak, whereas the clock goes literally flat, as you can see here. Whereas By giving high fat diet in combination with melatonin, that is intraperitoneal injection, it is somewhat restoring the peak over here. And same is over here also. It is restoring the peak, yes, but in the clock you can see a peak shift, as you can see here. Well, rest of the genes are showing some non-specific changes. Then talking about the circadian transcripts in the jet lag protocol, that is photoperiodic manipulation, uh, it is surprising to note that, like the high fat diet, even the photoperiodic manipulation shows a changes in the clock gene expression. But this is expected. But the previous one which I showed as a result of the diet is actually stunning. A combination of the two aligns more towards the high fat, high fructose diet. And as you can see here, the changes are taking place, the oscillations take place accordingly. Now, too much of peaks and ebbs and gene oscillations actually are confusing. That is why we do what is called as a circ wave analysis of the clock genes in liver. Now, circ wave analysis, it shows that which side of the graph are the dots aligning. All you have to do is just observe the black dots. Black dots indicates the normal position of the oscillation. And as you can see here, that especially the clock and the per is undergoing distortion, to severe distortion, as compared to, see, you can see here, it's supposed to be here, but it's going right at the extreme end. So there is an extreme distortion when it comes to the clock gene oscillations. Checking the same with the Western blots, the immunoblot analysis has been also extensively done, where the clock gene, as you can see here, is showing a peak shift. These are the two peaks of the clock, and there is a peak shift as a result of the high fat, high fructose diet, not corrected as a result of melatonin treatment also. So this is something which is quite significant. The same thing was subjected to circ wave analysis, and at the end of this, uh, one thing was clear, that exogenous melatonin, if given, if administered at the evening time, is able to somewhat restore the clock gene oscillations. Possibly that is the reason the metabolic function is restored to normalcy. All the parameters are not restored, but majority of them are restored. So at this point of time, we were very happy with the findings, and we went ahead with, well, this is another version of the analysis. But what, by this time, what we said, that melatonin is causing what is called as circadian reprogramming. All right. Now, the reprogramming of these clock genes is a big thing. And this, how is it doing? So, what we hypothesize, that it is possibly doing that in the liver through a central control, restoring the circadian disruption, altering the BMAL clock heterodimer formations, restoring it back to the normalcy, and restoring the oscillations of the subsequent genes. So this is the hypothesis that we had proposed, and we tested it and validated the hypothesis. And this was one of the very good recent publications that I got in FASEB, and the hypothesis that was proposed was published on the cover page of the FASEB journal. And at this point, we were very happy and ecstatic that we have been able to achieve a good study, and the finding is very novel and has been published in a journal which is very old and very highly reputed. People who are in this field of biochemistry, physiology, endocrinology are aware that this journal, biotechnology also, are aware that this journal is really very good. Well, but here at this point, something very interesting happened. When the animals were being observed in the cages, this was, Gopal sir, are you able to see the video?
यस सर दिख रहा है.
So this was the normal behavior of mouse. You can see here they are eating, and the jet lagged mouse, this was the behavior. They were all walking upside down. Now my student said, "Sir, can you just come quickly to the animal house? This is what I'm observing." We recorded the videos, and I said, "Well, this is weird. This is funny. Why is this happening? This needs to be investigated." But I am not a neurobiologist, but I said, nevertheless, we can give it a try. Let us check what's happening with the behavior. So we started investigating the mouse behavior, and there are set of experiments which you can perform. Like, you can study the emotional functions, you can study the cognitive functions and the motor functions. Emotional functions include tests like the elevated plus maze, open field test, tail suspension test, force swim test. Cognitive functions are also like the Y maze test, and motor function obviously is the distance that it walks in an open field. So to begin with, what we saw, well, we recorded the observations using a behavior, using a webcam, and then those that was played in a software, and the software could digitize it and then give us readings. So what will happen if there is a alteration in the circadian behavior? There will be easy fatigue. If there is a behavioral inhibition, then it's called as the psychomotor inhibition. Then there's something called as decline in socialness. We are also studying the social behavior, so not able to interact properly. Here, if we just take a pause and also think about ourselves, is our diet, is our sleep pattern affecting our behavior? Are we less social? Are we less motivated? Are we uh, generally showing a gross decline in our interest? Okay, then we are people who are in the high risk category of developing lifestyle disorders. Okay, talking about the whole board test. The whole board test indicates anxiety. There is a board, the mouse dips its head inside in the hole, and there is a beam which is below. And every time it dips the hole, head in the hole, the beam cuts and it is recording as one observation. So this was another one was the elevated plus maze test. This maze is such that on two sides there is like a wall kind of a thing, and two sides there is open arm. A mice which is having normal inquisitive behavior will be moving equally in all the arms. The mice that is anxious, डरा हुआ, डर रहा है, scared, will be spending more time in the arm which is having a wall. Now this is another very important test. You can see the representative data I have shown, animals data. So what we observed, that the mouse which was, uh, the elevated plus maze test showed that the high fat, high fructose diet fed mouse, it spent less time in the open arm and more time in the closed arm, indicating that it was very highly anxious. And by giving melatonin, it was sort of, the behavior was sort of restored, and it could spend equal time in both the types of arms. Another thing was that the high fat, high fructose diet mice, it spend less number of entries in the open arm. So number of entries were also counted, which were corrected by melatonin. And the head dipping behavior in the whole board test, as you can see here, both the high fat diet or the jet like mouse, it showed less number of counts, and exogenous melatonin was able to correct it. So one thing was clear: we have done not one but two tests, and both are uh, implying towards melatonin mediated correction of anxiety like behavior in mice. One more test, it's called as the marble burrowing test. A mouse will generally burrow anything that is foreign in its cage. Like here in this case, we have placed the marbles. Now, how many will it burrow? We have a normal, uh, you know, number, control, which is representing the normal number. Anything that is less than that, it means that the mouse is lethargic, the mouse is depressed. Anything that is more than that is the mouse is anxious. So this is what we found. We found that a lot more mouse was very highly anxious as a result of the marble burning. But here there was a flip side. Melatonin treatment also could not correct this particular aspect of behavior, right? So here, tentatively, what we can surmise out of this is that possibly melatonin is able to partially make corrections in the behavior, but not completely. Morris Water Maze is like a gold standard for learning, for spatial memory and learning, right? So there is a quadrant. People who are neurobiologists, they know this very well. But there is a quadrant. There's a stage, and the entire water is made white by adding milk powder, and the mouse swims inside. So the stage or the platform is removed. So how much of time the mouse is spending in that region where the platform is there. So if you divide this entire thing into four parts, how much time is spent here is what determines whether the mouse is learned enough, or is it still having a poor spatial memory. This is as a result of the high fat diet, that the spatial memory was poor, it had gone down. Whereas melatonin treatment to the high fat diet mice, it restored the memory. Same is for the test for depression. There is a forced swim test. A mouse which is depressed will, in a forced swim test, it will kind of give up. It will not fight. It will not struggle. But and also the tail suspension test, the mouse will kind of give up if it is depressed. But if the mouse which is normal will try and keep on struggling for a period of time which is under observation. What we observed was both the tests failed uh, in melatonin treatment, as a result of the melatonin treatment, because depression did not show any improvement as a result of melatonin treatment. So here what we can see is that it improves locomotor function, reduces excitability. To some extent, it is able to improve depression, but not totally. I will say just 5 to 10%, and it improves certainly the spatial memory. So exogenous melatonin, this is an unpublished data. Exogenous melatonin is able to improve these parameters. So the associated genes, we check the expression of the associated genes, the BDNF TRKB pathway, the SYN1, NT1, NT3, NT4, and all. So both the synapsin, PSD95 and all, they showed some restoration as a result of melatonin treatment, whereas there wasn't much of a difference that was observed in the immunoblots. Now, these two results, that is A, the RNA, and B, the immunoblots, these indicate one thing, that at the molecular level, possibly there are no significant changes that are seen as a result of high fat diet induced, uh, I mean, high fat diet, as a result of high fat diet. And exogenous melatonin is not able to correct whatever changes have occurred either. Here I was then thinking that, are we going on the right path? Because possibly the treatment that we have given has not manifested enough damage in the brain that is detectable. So I went back to the literature and found ample amount of literature which comprehensively said that even if the BDNF TRKB pathway is not showing a change, if the behavior is showing a change, it is considered as a change. That gave me confidence to move further in the study. So this study is over, but we have not published it yet. So in a nutshell, what we tried to say was that reprogramming of the core circadian oscillations in an experimental model of lifestyle disorder, it improves the neurobehavioral perturbations. As I told you that the understanding and studying the behavior was just an offshoot of the entire study. This also led to one more investigation, which was a very, well, this is a nice cartoon. It says uh, your biological clock is not ticking, right? So how do you assess whether you are having a problem with your biological clock? Okay, even till today, we do not have any method, and this is a question for all of you. Even I am trying to investigate. We do not have a kit, we do not have a test, we do not have a um, method of investigation or detection for our biological clock. We just rely on, yes, I do get a peaceful sleep or I don't. Yes, I am able to digest my food or I don't. I get hungry, I feel hungry or I don't. And we just rely on the indirect physiological evidences. That's it. So, talking about the epigenetic aspect of it, there is one RNA, microRNA. It's called as the microRNA 122, which is very liver specific. And guess what? Another student who was investigating microRNA found high levels of microRNA not only in the the function of microRNA 122 is it degrades the mRNA. So the student found high levels of microRNA 122 as a result of olic acid treatment, high levels of microRNA 122 in the high fat diet treated mice, as well as the chrono-disrupted mice liver, and also then we compared the circadian signatures of the hepatic and the serum microRNAs. And the serum microRNA is pretty much like a consistent level. Hepatic microRNA builds up and has a episodic release. Now, this was something which was, these are exosomal based release, obviously. So we are still in the process of finding out what can be the reason for it. So this is talking about the intra and the extra hepatic oscillations of microRNA, which you can see here that the in the case of liver there is a peak, whereas the serum it is flat, and these are, mind you, control mice. They get absolutely distorted in the high fat diet and the CD chrono-disrupted mice, as well as a combination of the two is also able to distort it. This is another ongoing study which we are yet to seek answers for that. So what I can sum up from my study is, I think I'm on time, which I am able to sum up from here is that the liver, there is some kind of a crosstalk that's happening with liver and some other organs in the body, and hence the microRNA 122 is being released. I hypothesize that possibly the microRNA 122 is received by the adipocytes, and that is why, as a result of giving melatonin, they get plump, because all the cholesterol and the triglycerides are taken up by the adipocytes. There is a pre-adipocyte to adipocyte differentiation. They multiply, they take up and clear the bloodstream, so that the animal is not at risk of any other secondary associated diseases. So I call it as a cry signal. Possibly the microRNAs are being leached out of liver to warn the entire body system. Let's not forget, our entire body is like a big co-culture, and these all factors, they'll be playing a role in the long run when it comes to a persistent low-grade inflammation, which is coupled with that. So here I will end my talk on a philosophical note, that this is the reason possibly that our great great grandfathers who understood the laws of nature revered the sun god as Bhaskar or Aditya, and there are lots of sculptures of Lord Sun God. There are sun temples. The Konark Sun Temple is very famous, and getting up early morning and worshipping the sun god, getting the sun rays on your body, triggering the biological clock, switching it on, and getting the metabolic physiology right is being greatly emphasized. Well, not only India, even in the ancient, say, civilizations like Babylon or Egypt, also you get the mention of sun god that is being worshipped and revered by the people. So now we are in the era of what is called as circadian medicine, right? Like, you take a medicine, but now the doctors are going a step further, or the researchers are advising the doctors to go a step further at what time the drug should be taken, because the efficacy and the effectivity of the drug, if it is having a circadian action, will vary on the time it is administered. Here I want to just draw your attention to the evening dose of melatonin that we had given to the mouse. Why evening dose? Well, this is, I think in 1986, there was a paper where a morning dose an evening dose and a morning and a evening dose was given to mouse, and it was found that the evening dose is most effective, obviously because mouse is nocturnal and high levels of melatonin in the case of mouse are at the night time. So that's why a evening dose was efficient. Then the other aspects are the drugging the clock. That is the therapies that are directly targeting the circadian clock that can correct the biological clocks, and then training your clock, that is maintaining a synchrony with the environment. That is the most important thing. That is going to bed on time, not getting exposed to the blue light, which disrupts the melatonin rhythm, even eventually the biological clock. This is my lab as of now. These are the three registered students. Rhythm Karnik is the Lady Tata Fellow. Shruti has submitted a synopsis and is writing a thesis. She is an ICMR SRF. And Heli Shah, who is a DBT JRF. She is the odd one out because she is working on the circadian basis of cancer. I've not worked with cancer till now, but then because of her interest in cancer, she persuaded me to take up cancer research. So she is
बहुत
working on this.
हार्ड वर्किंग स्टूडेंट है हेली.
Yeah, yeah.
डीबीटी स्पॉन्सर्ड उसने किया एमएससी, फिर आपके यहाँ गया.
I know, I know, I know. She has been a meritorious student and
हाँ हाँ
was a
हाँ
topper as well. And these are
हाँ.
my
हितार्थी भी मेरी स्टूडेंट बीएससी में, मैंने उसको किया था, उसका भी एक लेक्चर करवाया था. सी
जी
इज
जी.
ऑल्सो वेरी गुड. वो पोस्ट डॉक कर रही है ना यूएसए से.
Yeah, yeah. So
यस.
Hitarthi, I knew that will not talk about the
थोड़ा
circadian.
सा किया था सर्केडियन का, उसने थोड़ा
हाँ.
जेट लैग बताया था.
So वो थोड़ा मैंने overlap नहीं होने दिया.
यस सर, यस सर, यस.
So my recent so Ravi and Menka, Ravi Jadeja and Menka Thaunojam have been the uh, first two students. Then Kapil Upadhyaya is also there as a postdoc. Menka is a senior faculty. She is now an associate professor, and Ravi Jadeja is also professor, assistant professor now in the Georgia Medical School. Ali Asghar Vora was the recent one to join the University of Chicago at St. Louis. Kavita is a research intern right now. She was blessed with a baby. Deepak is in Malaysia. He's in industry as head of environmental toxicology, and other students have preferred to stay in India and are in teaching field, or Apeksha Joshi is in Sun Pharma. Recently, Apeksha Joshi has joined Jove, that is Journal of Visual Experiments, as a content writer. I acknowledge all the funding agencies. Well, GSBTM was my first grant, followed by the DST, then DBT, ICMR grants, and CSIR, DST, DBT grants is right now ongoing. And yeah, thank you very much for a patient hearing, sir.